Hello, my name is Philip Seitzer, and today I'm going to show you how to get started using the Comparative Genomics tool, JContextExplorer. First, to launch JContextExplorer, open an internet browser and go to the Facciati Lab website. The fastest way is to type Facciati Lab website in Google. Go to the software page and now click the large orange launch button. JContextExplorer is a Java WebStart, so as long as you have Java installed and running on your computer, you should be able to click the launch button to start the program. This is the opening window for JContextExplorer. You can see that the JContextExplorer context bar is the icon associated with this program. You'll notice that in addition to the main window, there's also a menu bar located at the top with numerous options. To begin using JContextExplorer, we must load in an appropriate data set. Datasets take the form of sets of annotated genomes, which are called genome sets in the context of JContextExplorer. There are several ways to load a genome set into JContextExplorer, but one of the fastest and easiest is to simply go to Genomes, retrieve popular genome set. If we look now at our current set of genome sets, we have none available. So let's load a data set. Again, go to Genomes, retrieve popular genome set, and today let's look at the Myxococcus data set. So we simply click on the menu item, and now we have a data set loaded. If we go back to Genomes, we'll see that now we have the Myxococcus data set loaded. We can also see information about this data set by selecting Current Genome Set or Command G from the menu. So here we see the set Myxococcus contains 5 bacterial strains, and we can find out information about the genomes located in this genome set by selecting them from the appropriate menu bar. Now that we have a set of genomes loaded into JContextExplorer, we can begin to conduct simple searches of the genomes. So let's search for all genes that have a gene annotation containing the word glucose. So we simply type glucose in the search bar with annotation search selected, and we can click Submit Search. A results window appears in the empty space located in the mainframe. We have our search, glucose. Each of these folders contains the name of an organism followed by a serial number, which is the instance that was found. So it looks like glucose is found in 13 different genes in the organism Anoromyxobacter dehalogenans. We can click on the folders and we can expand them to see their contents. We can click Expand All, see all of the individual gene instances, or Collapse All to hide them. If we want to filter this list further, down here in the Multiple Genome Browser Tool bar, we can type in filters. So let's say we want to select only the genes that contain the text string glucose from the organism Anoromyxobacter dehalogenans. I simply type that in. And we see that only these are selected now. We can also filter based on other qualities, such as annotation. Looking at the individual annotations, we see there is a great deal of variation in what these glucose hits actually are. For example, here this is D-TDP glucose 4,6 dehydratase. Here it's glucose 1-phosphate adenyltransferase. So let's filter, for example, for adenyltransferase. And now we see that only a few of these are selected. One of the major features of JContextExplorer is the ability to visualize multiple genomic segments simultaneously. To demonstrate this, let's select, in this case, just one genomic segment, and then here it is selected, and we can click View Contexts. If we do that, a window appears. The title of this segment is the same as the title here. As we can see, it contains only one color gene as well as several other grayed out genes. The color gene corresponds to this gene, and the surrounding genes show the context that from which this gene arises in the genome. If I click on genes, I can learn information about them. For example, here the annotation, the cluster ID, and the size. I can do this for both color genes and grayed out genes. If I can change what information I'd like to display on click. I can also change how things are displayed. For example, here I have Show Surrounding selected, but if I uncheck this box, all of the surrounding genes disappear. If I recheck it and then click Color Surrounding, now all of the genes have a color. If I check and uncheck Show Coordinates, I can see where in the genome this gene and genes are located. I can also edit the range that is shown around, in this case, this single gene. For example, let's say that I would like to look 20,000 nucleotides on either side. I click Update Context, and all of a sudden I see a much larger segment of the genome surrounding this gene. So far, I have only searched for single genes, but I would like to search perhaps not just for one gene, but for a set of genes, or for a gene and all the genes located nearby, or any number of variations upon a single gene search. I can do this by defining a context set. I can define a context set by clicking the Add Remove button under Context Set, or up in the menu under Load Context Set. So let's create a new context set. I'll call this one Range 1K. And what this will do, it will select, in addition to all of the query matches to my query, all of the genes that are located about 1,000 nucleotides before and 1,000 nucleotides after. I'll click Add, and then OK. And now in my drop-down menu, in addition to the single gene search, which will return only the gene, I now have a new context set, Range 1K, which will return the query match with all the genes located within a 1,000 on either side window. So let's repeat the search and see how things change. So first off, my original window is still here. I can have multiple frames, and I can click between them, minimize and maximize and so on, and resize as well. So let's go back to the original, and let's select all of these queries that also have a cluster ID number of 1011. I do that by typing in all caps cluster ID : and then the number 1011. I discover that this particular gene, remember here I'm in single gene, so let's just start with 1. Here it is. And let's see how that looks when I am looking at not single gene but 1K. So I can see that now, in addition to this gene, I'm including these two other genes, one on either side, because those are within a 1,000 base pair window of my query match. So let's go back to this selection of cluster ID 1011. These are all of the genes that have a cluster ID of 1011 within the subset search glucose. So as you can see, there are 6 contexts in total, and this blue gene in the center is cluster ID 1011. Genes are always color-coded according to commonality. In this case, we searched for glucose, so we had an annotation search, so everything will be colored based on common annotation. You can select multiple groupings by right-clicking or control-clicking if you're working on a Macintosh, and clicking Show Legend Complete. This brings up a window that shows you the mapping of colors to annotations to cluster IDs. So here, I was interested in cluster ID 1011. I can select it in this window and I see that every instance is now selected in my browser window. I can select multiple using Shift and Control clicks. Now I'm interested in not just visually observing how those differences in that cluster looked, but also mathematically describing them. So I'm going to create a new search. I'm going to switch to cluster number, and this time I'm going to search for all instances of cluster 1011. And I'm going to add, in addition to printing the search results, I'd like to render a context tree. And I will again make sure that I am working with context set range 1K. So now, as you can see, we have the results, cluster 1011. But now we have a new frame, the context tree, and we see is a very simple tree showing the relative relationships of these contexts. So you can select the individual leaves of the tree by clicking on them. You can select a group by holding down Shift when you click. And let's take a look and see these two groups, which are very closely related. Let's see what those contexts look like. So as we can see, they look very similar, so it's no surprise that they would be grouped very closely together. Now if we compare that to, for example, this other one, which is further away, we see that now there's a different gene here, so it's no surprise that these are further away, that there's some distance there. Note that when I select things in this window, they also appear selected here. And similarly, if I select them in the search results window, you can see them in the context tree. Our usual text-based selection also works. For example, here I've selected all of the Myxococcus, and once again, those are selected in all windows. This completes the introductory tutorial for JContextExplorer. Please check out the user's manual, which you can reach by Help User's Manual, as well as the additional tutorials. Thank you.